Now we are going to discuss regarding the type of bioreactor used. The type of bioreactor that will be used in the study is the fibrous bed bioreactor, or also can be known as FBB. In this study, a fibrous bed bioreactor is preferred as the process requires cell immobilization for optimum output. Fibrous bed bioreactor that is being used is a system with medium circulation made out of a jacketed glass column that is packed with spiral wound fibrous matrix, and it is run under the continuous mode. So this is the design of the FBB. For pilot scale, it is made up of glass and it has the recirculation pump, tube for medium addition and harvest, water jacket, fibrous matrix, and water inlet and outlet. For the experimental setup, it is run under continuous mode in anaerobic condition, hence explain why nitrogen gases are being purged instead of oxygen. And this table shows the specifications of the FBB in industrial scales. In industrial scale, the design type is vertical and made up of stainless steel. For the FBE fermentation, the upstream processing will involve two phases, which are acidogenesis and solvatogenesis by Clostridium acetobutylicum and Clostridium thyrobutylicum. So for the upstream processing, the size of the fermentation will be scaled up gradually. It started with the inoculum preparation before it is transferred into the lab scale at 1 liter, and for pilot scale, it will start at 5 liter, 50 liter, 100 liter, and for the industrial, it will involve two fibrous bioreactor. The bioreactor will specifically use for acidogenesis and solventogenesis phases. So what is acidogenesis and solventogenesis? The dilated conciliary and PT medium is fed as nutrient supply to the bacteria. In acidogenesis, the Clostridium thyrobutylicum will consume the nutrient and produce volatile fatty acid, which is butyric acid, lactic acid, and acetic acid. While for solventogenesis, the operating condition for the second phase bioreactor is exactly same as the bioreactor in the first phase, except that the strain that has been used or immobilized is the Clostridium acetobutylicum. And at this stage, it will not produce any volatile fatty acid. Instead, it will convert the butyric acid into solvent form, namely butanol, acetone, and ethanol. For SuperPro Designer, the first two units involved are the seed fermenter or breeding tank 1 and 2. It serves as holding tank and to grow each species of the bacteria to a set number before immobilization and fermentation stage. The next unit operation involved are acidogenesis and solventogenesis fermenter. To run for the acidogenesis phase of fermentation, Clostridium thyrobutylicum are used and the contents are pumped in the first phase fermenter from breeder tank 1. For solventogenesis FBB, it is run in parallel. Clostridium acetobutylicum are inoculated in these fermenters. Fermenters are kept at 37 degrees Celsius and more acidic pH, which is also controlled by buffers. And at this stage, glucose are utilized and produced into acetone, butanol, and ethanol. The last unit procedure for upstream processing is the heat sterilization. The emission from the solventogenesis will be flows into the heat sterilization unit. The temperature will be set at 131 degrees Celsius and duration will be set at 30 minutes.